Hello, everyone. This video is going to look at how to do some basic data analysis using Microsoft Excel. We'll look at how some pretty simple calculations can be done and also how we can plot some of the data to make some useful graphs that present it. The first data set looks at the vegetation cover of shrubs in a mountain range in the Mojave Desert, measured across a 50-meter transect. And so in the first column of this data frame, we see each of the species. And in the second column of this data frame, we see the number of centimeters within that 50-meter transect that each species occupied. So for example, Frangula californica occupied 1,065 centimeters within this 50-meter transect. Quercus turbinella occupied 2,265 centimeters within this 50-meter transect. Pinus monophylla occupied 650 centimeters within this 50-meter transect, et cetera, et cetera. And what we want to do with this data is use it to calculate Shannon's diversity index. And as you might recall, Shannon's diversity index is basically a measure of diversity that counts for both species richness, that is the number of species, and species abundance, how many centimeters within that 50-meter transect did each species occupy. And as you might recall, Shannon's diversity index is calculated as the sum of proportional cover times the natural log of proportional cover of each species in the particular data set. So how do we go about doing this with this data? Well, it's pretty straightforward. We would basically take the number of centimeters that a particular species occupied within the 50-meter transect, which in this case with Frangula californica is 1,065 centimeters, and we would divide it by the total number of centimeters that the transect covered, which is 5,000 centimeters, which is the same as 50 meters. And so we can do this for each of our species. So for example, here in Excel, I would take the value of centimeters for Frangula californica in this column B and this row 2, so that is B2, and I would divide it by 5,000. And in fact, rather than having to do this over and over again for each species, what you can do is simply take this value and divide it by 5,000 in this way. Make sure you put an equal sign every time you try to do that. And you'll get the number 0.213. That is the proportional cover of this particular species in the transect. And if we want to do that calculation for all of the species in the data frame, rather than doing it over and over and over again, once we've done that calculation one time, all we have to do is grab the bottom right of this little cell, and you'll see this plus sign emerge. And you can click on the bottom right of that cell with the plus sign and drag down, and lo and behold, all the calculations of proportional cover will be done for you. So this happens pretty fast. It's pretty efficient. Likewise, if we want to calculate proportional cover times the natural log of proportional cover, we could take the value of proportional cover and multiply it times the natural log of proportional cover. Basically, the way that we would do that is by taking this value here, this value right here. So start with the equal sign. We're going to say that proportional cover times the natural log of proportional cover equals the proportional cover here in this particular cell times parentheses natural log, which is a function in Excel characterized or symbolized by ln. So ln is a function that Excel recognizes. ln natural log of parentheses proportional cover, close the parentheses, and close the parentheses one more time to complete the expression. And now Excel will calculate proportional cover of the species times the natural log of proportional cover of that species. You hit enter, and we have the value negative 0.3294 as we see over here. Likewise, if we want to do this calculation for all of the other species in the data frame, rather than do that over and over again, we click on the bottom right corner of the cell, get that plus sign, and you can drag down, and lo and behold, we have all the values of proportional cover times the natural log of proportional cover. And recall that Shannon's diversity index is the sum of all these values times -1. So basically, if we want to get Shannon's index, all we have to do is go to another cell, enter equals, and say sum, which is yet another function that Excel has. And tell Excel you want to sum up all of these values. And you do that by saying equals sum parentheses, all of the values, you highlight them, close the parentheses, and multiply, which is a function signified by using an asterisk in Excel, multiply by -1, put -1. It'd be wise to put -1 in parentheses. I don't think we have to, but for sake of tidy data, we can put it in parentheses. And there we have Shannon's diversity index calculated for us, which is in this case equal to about 1.32. So this is a pretty simple example of how to take data collected on vegetation cover in a transect and use it to calculate diversity. Now you could do this with all kinds of other data, not just vegetation cover in transects. There are all kinds of ways you can collect different kinds of data across different surveys that you might be doing and use it to calculate diversity. This second data frame that we have here looks at life table data collected from this annual plant called Phlox drumondii. And as you might recall, basically a life table includes information about the number of individuals within a cohort over a particular course of time. In this case, we're basically tracking this cohort of Phlox drumondii for about a year or one growing season. And what we have in our data frame is basically this thing X, which represents different periods of time that we took measurements on this population of Phlox drumondii, and this thing called A sub X, which represents the age classes that we're taking measurements. This thing called N sub X, which represents the number of individuals in the population at a particular age class. This thing called L sub X, which represents the survivorship of individuals in this population at different age classes. This thing called big F sub X, which represents the number of offspring this cohort may have produced at a particular age class. Something called fecundity, represented by M sub X. And then these two other variables, LX, MX, which represents per capita fecundity and the sum of age class and per capita fecundity. So basically what we see is that data was collected on this population at a different points in time. The first data that we have comes from 63 days after this population first emerged. That is, all of these little seedlings started to germinate. And after 63 days, we can see that there were 996 seedlings that survived in this population. We can see that after 124 days, there were only 668 seedlings that survived. After 184 days, there were only 295 seedlings that survived, so on and so forth, all the way throughout the course of 363 days, at which all of the seedlings had actually eventually died. The first kind of calculation we can do here is survivorship. And in order to calculate survivorship, we're basically saying survivorship represents the number of individuals alive at a particular age class divided by the number of individuals that were alive at the first age class. And so in this particular case, in the first row of data we would take the number of individuals at 63 days, which is equal to 996, and divide that by 996, which is, of course, equal to 1. For the second row of data that we have, we would take the number of individuals that survived at the second age class after 124 days and divide that by the number of individuals we had at the first age class, which is equal to 996. And we can see that that value is not equal to 1. It is instead equal to 0.67. Now, before going too much further, what I'd like to point out is that in order to do this kind of a calculation, it would make your life a lot easier to use the dollar signs to hold the denominator of this calculation constant for all of the calculations. So just for sake of example, if we were to be doing this from scratch, I would say that the value of survivorship equals the number of individuals at this age class divided by that same number of individuals, but by putting a dollar sign in front of the specified column and a dollar sign in front of the specified row, we can now make sure that that value is always the one that's in the denominator as we make our calculation of survivorship. And as we sort of saw before, rather than doing this calculation over and over again, we click on the bottom right corner of this cell and drag down all the way through all of our rows. And we can see that all of the calculations for survivorship will be made for us pretty easily. So this is a very convenient way to calculate survivorship. The key here is making sure that you put those dollar signs in front of the column and row that you are putting in your denominator. Okay. Then we get to this point in time where we have some data on the number of offspring that are being produced. And basically, if we want to calculate fecundity, we know that fecundity is the number of individuals that exist at a particular point in time divided by the number, the number of offspring that exist at a certain point of time divided by the number of individuals. So this is pretty simple. We say, okay, we have 53 offspring at age class 7. We're going to divide that number of offspring by the number of individuals in the population, which is 159. And likewise, we would do this for all of the other instances where there were offspring. So for example, if we want to calculate fecundity, we would say that this value of fecundity equals 53 divided by 159, and we get 0.333, representing fecundity at this age class, which is the average number of offspring produced per reproductive individual. And likewise, if we want to do this for all of the age classes where we have offspring, rather than do the calculation over and over again, we drag down, and now we have our fecundity values for each age class. Trying to get the product of survivorship and fecundity is relatively straightforward. We're simply going to take the product. And in order to do that, we would go to the cell here, enter equals survivorship. Remember the asterisk in Excel is the product function. This is multiply by. So the product of survivorship and fecundity is equal to the product of these two values. And there we have that value, 0.053. And again, if we want this value for all of the age classes, we simply drag down. There we have it. Likewise, if we want to get the product of age class, fecundity, and age class survivorship and fecundity, rather, we take the value of age class from here. This value equals age class 306 times per capita fecundity. And we have the value there. And again, if we want to get this value for the age classes where the data is relevant, we drag down, and now we have all of the data values. This is very efficient. The two kinds of interesting parameters we would like to get out of this data set are net reproductive rate and approximate generation time. As you might recall, net reproductive rate is simply the sum of per capita fecundity. And we have that right here. And as you can see, I took the sum of all of these values here, and I was able to calculate net reproductive rate pretty quickly. So just to show you how you could do that, find a cell equals sum all of your per capita fecundity values up, and there we have net reproductive rate. Approximate generation time, as you might recall, is the sum of age class and per capita fecundity divided by net reproductive rate. So we can see how I made that calculation here. I summed all of these values up and divided them by net reproductive rate. We can do this again, for example. So in this cell, we say equal sum of product of age class and per capita fecundity divided by net reproductive rate. And we get an approximate generation time of 337.3 days. And so these values are telling us, first of all, net reproductive rate on average. This is how many offspring an individual was producing during this particular period of time. 2.4 offspring on average per individual were reproduced. And on average, individuals were reproducing at around 337 days. So this is useful information. We can also look at how we might be able to graph this if we want to look at how population growth is changing. So we could compare time on an x-axis to population size on the y-axis. So basically what we would do here is take data for the points in time when population size is actually increasing, and that would be the points in time when this population is producing offspring. And that turns out to be at age classes 306, 320, 334, et cetera, et cetera. And we would look at the number of individuals that the population has at that particular age class. And slightly trickier to look at this, but the idea is we want to think about, well, how many individuals are there uh, who are reproducing and how many offspring are there? And at this age class number 7, that turns out to be 159 plus 53. At age class 8, which is after 320 days, we see that the number of individuals equals 154 plus 485 offspring plus the other 53 offspring that were produced in the previous age class. Likewise, when we go to age class 9, we see that the number of individuals is equal to 147 plus 802 offspring produced in that age class, plus 485 offspring produced in the previous age class, plus 53 offspring produced in yet the other previous age class, etc. And in the next age class, the number of individuals is going to be 105 plus 973 offspring, plus 802 offspring, plus 485 offspring, plus 53 offspring, etc. I think you see where this is going. And in the final age class where reproduction is happening, the number of individuals is equal to 22 plus 95 offspring, plus 973 offspring, plus 802 offspring, plus 485 offspring, plus 53 offspring. And so this gives us a way to look at how over time this population size has increased. And we can plot this data pretty simply here in Excel. And so the way you would do this would be to take these two columns of data, and you can highlight them like this and go to Insert. And Excel has all kinds of different charts that you can make with it, and you kind of got to be clever about knowing which is the right kind of chart to make to represent your data. In this example, what we want is a line chart based on a scatter plot, because we have a scatter plot of continuous data on both our x and y axes. Now you can make a scatter plot like this. I think it's a little bit more informative to make a scatter plot that has a line in this example that kind of helps illustrate how population growth is increasing. So that's the way I chose to do this. So again, in order to produce this plot, I simply highlighted my two columns of data, the x-axis represented by age class, the y-axis represented by population size, and I selected a scatter plot with a line. And that gave me my graph. Now, a couple tips I'll give you on making nice graphs. First of all, chart titles are usually always useless, so I would delete that. And lines within the matrix of the plot are kind of useless too, so you might delete those. Kind of up to you aesthetically how you want to do it. You might have a plot like that. I guess I left the lines on that plot. So whatever, I guess we'll leave them. But the other thing that's really important is to label the axes, because without labeling the axes, we have no idea what this graph shows. Labeling axes is not that difficult. You click on chart design, which is already highlighted here, and go to add chart element, and we have axis titles, and we have the horizontal axis title and the vertical axis title. And so you could simply click on horizontal axis title, and here you can enter in what this means, and it is age class presented in units of days. So it's always important to note what units your axis is representing. So we have age, class, and days. And then we can go ahead and label the y-axis, the vertical axis, and this is population size, is denoted as n. And now we have a relatively nice plot that shows how population size is increasing kind of exponentially over this period of time that we have data for. As you might notice, I like to make my plots relatively more square in shape. Typically, if you're trying to publish a figure, journals will want stuff to fit inside the body of text very nice and tidily. So a square plot is often nicer than a rectangular plot, which you might get by default. So like, for example, when you first say make a line plot, it makes it kind of rectangular. You can change that kind of as you see fit by just moving around the dimensions of the plot. So that's kind of aesthetically a decision for you to make. So we have a nice plot that shows how population size in this particular example has increased over time. We can also take this data to calculate how the rate of population increase is changing over these various age classes. And as you might recall, this is basically done by taking the product of the intrinsic rate of population increase and population size. The intrinsic rate of population increase is equal to the natural log of net reproductive rate divided by approximate generation time. And so that calculation is pretty easily made. I can do it for you over here by saying, okay, R is equal to R is equal to the natural log of net reproductive rate divided by approximate generation time. And you need to be very careful with your parentheses here because Excel will not calculate things correctly if the parentheses are not correct. See, you already got mad at me. That is our intrinsic rate of population increase at every age class. All we need to do now is multiply it by population size at the age classes of interest. And we get product of our intrinsic rate of population increase and population size. And we can look at this at every age class, again, by clicking on the bottom right corner of this cell and dragging down for all the age classes. And now we can see we have a problem. I'll just show you here. What we can see is that at each age class, the rate of population growth is sort of increasing and increasing in an exponential way until it sort of finally levels off at the end. And the problem here, the reason why I got a little error was because I forgot to put my dollar signs that tell Excel to always use these particular cells to make the calculations. So what you'd want to do is put a dollar sign in front of your particular column of interest and row of interest. Likewise, column of interest, row of interest. This is telling Excel to always use these particular cells as values that will be included in this calculation. And so now I should be able to drag down, and lo and behold, I will have the product of intrinsic rate of population increase and population size for each of the age classes I'm interested in. And this data here, these calculations we made, really corroborate well with what we see in the plot. Like population size is increasing a little bit at first and a lot more later on, and then again sort of leveling off toward the end of the period of time that we're looking at. This next data frame looks at some different transects of vegetation cover that were conducted in mountains of the Mojave Desert and in mountains here in the transverse ranges of Southern California. And there were 7 transects done in each site. So we see these 7 transects, and we have measures of Shannon's diversity for each transect. So for example, here in this mountain range in the Mojave Desert, this is Shannon's diversity for one transect, Shannon's diversity for another transect, et cetera, et cetera. And in the transverse ranges, this is Shannon's diversity for one transect, Shannon's diversity for another, et cetera, et cetera. So we have 14 transects in total. And what we want to do with this data is use it to calculate the mean of diversity for each of these environments. So we want to know what is the mean diversity of transects in mountain ranges in the Mojave Desert, and what is the mean diversity of transects in the transverse ranges. And we also want to use this data to calculate the standard deviation, which is a measure of variance among these different transects. And this is really pretty straightforward. If we want to calculate the mean of diversity in this mountain range in the Mojave Desert, we would simply say that this set of values is equal to this average. So for example, you would enter equals into a cell and write the word average. Average is a function in Excel, and it will ask you for the data, and you would enter all of the data in like this, close the parentheses, and Excel will calculate the mean diversity for these 7 transects. You could do this again for the other mountain range, but actually a neat trick is that instead of doing that all over again, once this calculation has already been done in a particular column, you can click the bottom right of that calculation and drag it over, and it will calculate mean for the adjacent column. So this is a pretty quick and efficient way to do this. Likewise, if you want to do standard deviation, there is the standard deviation function in Excel. Pretty straightforward. You'd click on a cell, you'd enter S, E, 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 E. That's the function. And you tell Excel which values do you want to calculate standard deviation for. Highlight them all, close the parentheses. There we have. Sorry, I found a small error. So there we have standard deviation calculated for our 7 measures of diversity at this mountain range in the Mojave Desert. Likewise, instead of doing this calculation all over again, you can drag this uh, cell to the right, and it will calculate standard deviation of the adjacent cell for you. So here in just a few moments, we've been able to take data, which it took a long time to collect this data. Doing 14 transects takes hours and hours and hours, but here we can calculate mean and standard deviation of our data in just a couple minutes. Second thing we want to do with this data is use it to perform a 2-sample t-test. And a 2-sample t-test, as you might recall, is going to compare the mean and variance of these 2 data sets. So how much of the difference in the means between these 2 sets of data can be explained by the variance among them. And so this is effectively telling us whether or not diversity in these 2 sites is significantly different. Is the diversity at this mountain range in the Mojave Desert higher or lower on average than diversity here in the transverse ranges of Southern California? And since we have replication, because we've done 7 transects in each of these mountain ranges, we have the ability to use that replication to answer that question statistically using something called a 2-sample t-test. In order to do a t-test, there are a couple things you might have to do. I have already done them. I have the data package. Get this out of the way so you can see. I have the data package, data analysis package already loaded in Excel. You might have to do that if you haven't already in order to access the automated functions that allow you to do this. But once you do have that, you should have this little thing in the far right when you click on data that says data analysis. And when you click on data analysis, you'll see that there are these different analysis tools. Excel can perform a number of different statistical analyses. And what we want to look at is a 2-sample t-test assuming either equal or unequal variances. I'll show you both. It kind of doesn't really matter which one you choose. You'll see that the result's not going to be terribly different. But let's say we choose to do a t-test, a 2-sample t-test, assuming equal variances. We click OK. It's going to ask us which variables we want to analyze. And because this is a 2-sample t-test, it is analyzing 2 different columns of data. And not surprisingly, the first column of data would be our diversity values from the 7 transects in the Mojave Desert mountain range. And our second column of data would be the values of diversity from the transverse range transects. And importantly, recall that when we're doing a t-test, we are testing a null hypothesis. And the null hypothesis is that the difference between the mean, in this case, the mean diversity of these different transects is equal to 0. The alternative hypothesis is that the means are significantly different. So the point is that when you're running this analysis in Excel, it's going to ask you what the hypothesized mean difference is. And in our scenario here, the hypothesized mean difference is equal to 0. It's also going to ask you what alpha value is, and this is basically where we're setting the threshold of statistical significance, which, as you might recall, conventionally speaking, is usually going to be 0.05. This is the probability of rejecting the null hypothesis by accident. So we're saying we're willing to accept a 5% chance of rejecting our null hypothesis by accident. This is, by definition, the probability of a type 1 error. And then lastly, it asks you, where do you want this output to be presented to you? Do you want it presented on the sheet that's in front of you? Do you want it presented on a different sheet? So I guess for now, we'll say, let's present it on a different sheet. And pretty simply now, you simply click OK. And here we have our results shown on a different sheet. You might want to expand it out because it doesn't do that automatically for you, so you can read all of the things going on here in the results. And this is very, very nice. It calculates the means for you. And variable 1, by the way, is the transects from the mountain range in the Mojave Desert. Variable 2 represents the transects from the transverse ranges of Southern California. So we have our means calculated for us. And as you might notice, they're the same means that we calculated here. We have the variance calculated for us. And if you don't realize this, standard deviation is the square root of variance. So if you were to take the square root of these two values, you'd see that they equaled the standard deviation that we already calculated. We have the number of observations in each category, 7 and 7. Blah, blah, blah. We have our degrees of freedom. This is pretty important. And the degrees of freedom, as you might recall, is a penalty we take for subsampling because we didn't sample vegetation everywhere. We did 7 transects in one area and 7 transects in another that represent the larger population of all vegetation cover amongst these areas. So we take a little bit of a penalty to sort of give us a little bit more of an accurate statistical measure. And in this case, the degrees of freedom is the number of observations in each category subtracted, or I should say the number of observations in each category minus 1. So 7 minus 1 is 6, 7 minus 1 is 6, 6 plus 6 is equal to 12, or in other words, you could say 14 minus 2 is equal to 12. So those are our degrees of freedom. We have a t-test statistic that is very nicely calculated for us. As you might recall, t is a kind of complicated statistic to calculate by hand, and so Excel can do it for you, saves a whole lot of work. And then down here we have our p-values and these things called critical values. And basically the critical value is the value at which if t is equal to the critical value, then probably I should say if t is equal to the critical value, then it means that probably it's going to be significant. I want to rephrase that. If t is equal to or less than the critical value, then our p-value is going to be less than 0.05, which was the alpha threshold that we set. There's some information here about tails. It might sound weird, but basically this is testing tails of a normal distribution. So if you think about that bell curve, it has tails at the end, at the lower and higher ends of the distribution. And it's kind of telling us that we can test our data using one of those tails or both of those tails. We really want to use both of those tails because that gives us a bit more statistical power. And so we would look at our p-value with 2 tails, and we would look at our critical value with 2 tails. And our critical value is way larger than our t-stat, and so that tells us right away that the results are significant. That is to say that the means of the two groups are really significantly different than one another, or sort of significantly different than one another. Our p-value is equal to 0.065. Which is actually a little bit above our alpha value, but we would call this marginally non-significant. So in this case, there is a 6.5% chance that we reject our null hypothesis by chance. I also did this analysis using a two-sample t-test, assuming unequal variances, which our variances are not exactly equal to one another. But we could do that test using the same methodology, and we would see that the results would be quite similar, not exactly the same. The t-stat is going to be the same. So we see minus 2.02 is our t-stat here. Likewise, minus 0.2, minus 0.203, I should say, minus 0. Sorry, minus 2.03, minus 2.03 is the t-stat, regardless of which analysis we use. But the critical values are going to be a little bit different, and the p-values are going to come out to be a little bit different. And in fact, we see that our two-tailed p-value using a t-test that assumes unequal variances is slightly higher. So this tells us that there's a roughly 7% chance that we would reject our null hypothesis accidentally. So this actually, doing a t-test with an assumption of unequal variances actually gives us a little bit more statistical power. But as you can see, the results are not terribly different. And so how do we interpret this result? Well, we would say that there is a marginally non-significant difference between the means. Probably there's something going on. If I was to interpret this, I'd say that diversity seems to be a little bit higher in the transverse ranges of Southern California compared to these transects we did in mountain ranges, this mountain range in the Mojave Desert. If we had more data, that might change the results, and we might see that the p-value would be closer to 0.05, or maybe not. Maybe the p-value would be higher if we had more data. So it's kind of hard to say. This is why we kind of want to look at standard deviation, and this gives us kind of an idea of how much variance there is in these two data sets. And a great way of looking at this is to graph the data. How do we go about graphing the data? Well, we can produce a graph like this pretty quickly if we simply say, well, here are our two categories which represent the independent variable. The two different mountain ranges represent our independent variable in this situation, and diversity, or rather mean diversity, represents the dependent variable. So if you wanted to graph this, you'd select these two headers for these two categories of mountain ranges, and then push Control on your keypad, and then select values of means for those two categories. And this will tell Excel to link these items, and then go to Insert. And instead of clicking on a scatter plot as we did before, we want to click on the bar plot option, which is here. And we can pretty quickly make a bar plot. And our bar plot is sort of already nicely labeled with our two mountain ranges and our mean diversity values. All we need to do with the bar plot is clean it up a little bit. Again, chart title is kind of useless. I would delete that. I think these lines are kind of useless. I would delete them. You can click on these lines and you can delete them. And as I mentioned before, it's kind of nice to present a plot like this in more of a square, in more of square dimensions rather than rectangular dimensions. So I've gone ahead and done that. That is aesthetic, and that's kind of up to you. And then we need to label this y-axis because we don't really know what all of these values represent. So we go to Add Chart Element, Axis Title, Primary Vertical, Axis Title, and we can enter in here what that axis represents, and that is diversity. And now we have a really kind of nice bar plot. How do we go about adding the error bars? If you notice, the graph I made has these error bars, which represent standard deviation for both of the data sets for both of the categories of mountain range. Well, this is slightly trickier, but certainly doable. We go to chart element, error bars, and it gives you some options. I'm going to tell you right away that Excel, whoever programmed Excel, didn't know a whole lot about what error bars are supposed to represent. So these options here don't really tell you anything very meaningful or anything very accurate. What you want to do is go to more error bars options, format error bars, and go to the very bottom. They just hide this from you. I don't know why. And select custom. And once you select custom, you can specify the value that should be added to these error bars. And what you can see is you have the opportunity to enter that information in. Where are you going to get that information from? We already calculated it. What we're going to do is say the positive error value for both categories, positive error bar values for both categories are equal to their relative standard deviations. So you can highlight the two columns or two cells of standard deviation for both categories for the positive error bar value. And likewise, do the same thing for the negative error bar value. Be very careful about this. Double check, make sure that these two things you've entered in are the same, because you want the positive error bar value to be the same as the negative error bar value. And you want to make sure that these error bar values are representative of the different standard deviations for each category. Once you click OK, we can see that we now have our error bars. Now, another thing I'll point out with this plot is it's kind of hard to see the error bars because the bars are blue, a dark blue, and the error bars are black. So you can click on the bars themselves and right click, and then you see the option to change the color of the outline. And you can use whatever color you want. I recommend using a color that makes this plot, sorry, not outline, fill. Click on fill, and you can choose whatever color you want, but I recommend using a color that sort of makes it easier to see the error bars. And so I usually would pick something like a light gray, like this. So again, this is fill, not outline. And that changes the color of the bars so that now we can clearly see the error bars with the means. And now what we have is a graph that shows bar plots with means plus or minus standard deviations of these two data sets that very much reflects the results of our two sampled t-tests. And it's nice to think about this. The standard deviation for the data set from the mountain range in the Mojave Desert was 0.37. And we can see that the mean plus 3.7 is represented here, and the mean plus, I'm sorry, the mean plus 0.3.7 is represented here, and the mean minus 0.37 or 0.3.8 if you round is represented here. Whereas the standard deviation for the transverse ranges of Southern California was equal to about 0.23. And we can see that that's true here. This error bar represents the mean plus 0.23 or the mean minus 0.23. And so the error bars are not the same. If you had allowed Excel to automatically calculate the error bars for you, even if you would have said standard deviation, it would not have done it correctly. It would have given you some arbitrary values. So this is the accurate way to go about doing this. As you can see, the way we made this plot is very much the same way that I made this plot. And then the last data frame that we have is basically one that we can use to do a chi-square analysis. This data frame looks at counts of birds that are categorized by their feeding guild or their feeding strategy at a number of transects that were conducted in the Verdugo Mountains here in Southern California. Some of which were conducted before there was a wildfire. The Latuna fire actually in 2017 is the fire we're talking about, and some of which were conducted after the fire. We have 6 transects that were conducted before the fire and 6 transects that were conducted after the fire. And we have the counts of birds who either tend to prefer to feed on seeds or those that tend to prefer to feed on insects, those who tend to feed on nectar, these are hummingbirds, those who tend to feed on carrion, so things like vultures or hawks, or things that just tend to be generalists that will feed on anything. So we have these 5 different categories of bird feeding guilds and the counts of birds that were observed in the different transects before the wildfire and after the wildfire. What I've done here is gone ahead and added up these counts for the post-fire data and the pre-fire data. And so that was basically done by saying the sum of birds that were eating seeds, or rather the sum of seed-eating bird species that were counted on the post-fire transects is equal to all of these values summed up here. So the sum of items from column H in row 2 to column M in row 2, and likewise the sum of counts of birds that are seed-eating species that were counted during the pre-fire transects are being summed up by the sum of values in column B in row 2 to column G in row 2. And I did that calculation for all of these different categories in order to get the total counts for all of these different birds in these different categories, whether they were counted in pre-fire transects or post-fire transects. Here we have what's basically called a contingency table. And we can use this contingency table to do a chi-square analysis. And as you might recall, if we want to do a chi-square analysis, we're basically comparing observed and expected observations. And so for this particular analysis, I went ahead and said, I'm going to call the post-fire count observations my observed data, and the pre-fire count observations the expected data. So we're kind of saying, we're expecting that the fire didn't do anything to the proportions of these counts. And that's sort of like testing the null hypothesis that there's no difference between observed counts before the fire and observed counts after the fire, as if to say the fire had no effect on the counts of birds who belong to these different foraging categories. So again, we're calling our pre-fire counts our expected data, and we're going to test that expected data against our post-fire counts, which are our observed data. And again, as you might recall, chi-square is calculated as the sum of observed minus expected values squared divided by expected values. And not surprisingly, we can do that calculation pretty easily here in Excel, and that is what I've done here. So I've basically taken my observed values minus my expected values for the guild of seed foraging birds, squared them and divided them by expected values. Again, I would point out you need to be very careful with the parentheses here, because if you don't really classify your calculations properly within the parentheses, they might not be done correctly. So anyway, I've gone ahead and calculated that observed minus expected squared. By the way, I'll point out that squared in Excel is calculated as caret 2. As a matter of fact, I'll just show you how you can make this calculation here. If we wanted to do this again, the values of observed minus expected squared divided by expected would be calculated as equals observed minus expected. Caret is like saying squaring this thing, power of 2, and put that whole expression, the Excel gets mad very fast. Put that expression in parentheses and divide it by the expected values. Excel is just very mad at me right now. Let's do it again. So this equals observed values minus expected values squared, close the parentheses, divided by expected values. And we get that particular statistic. And again, as we saw before, rather than doing this calculation over and over again, we can click on the bottom right and drag down. And now we have this value calculated for each of our 5 categories. And as you might recall, the chi-square test statistic equals the sum of those values. So this is pretty easy to calculate by telling Excel in this cell equals the sum of these values. And now we have our chi-square test statistic. What do we need next? We need to know what the degrees of freedom are. And as it turns out, you might recall, the degrees of freedom for a contingency analysis equal the number of rows minus 1 times the number of columns minus 1. In this case, we have 5 rows and 2 columns. So we can calculate the degrees of freedom here as being equal to 5 minus 1 times 2 minus 1, and that gives us 4 degrees of freedom. We're up and rolling. All we need to do now is calculate our p-value. The p-value can be found by using a function called chi-square distribution. So if you type CHISQ.dist into Excel, it has a function that will calculate the chi-square distribution. And it has some arguments that need to be entered into the function. The first argument, x, is the chi-square value. The second argument is the degrees of freedom. And the third argument has to do with which distribution you want to test this against. So we basically would enter in our first argument, our second argument, and without going into nitty-gritty details, I'll just tell you that third argument needs to be called false. Once the 3 arguments are entered into the function, you hit enter, and we have our p-value calculated for us very nicely. And as we can see, our p-value is way, 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 way less than 0.05. So it appears that there is a very significant difference between our observed and expected observations of counts of birds in these different feeding categories, which is effectively really telling us that as a result of this fire, maybe there's something going on in the environment that has caused the different proportions of feeding guilds of birds to have been changed, probably because the structure of vegetation in this environment has really significantly changed as a result of the wildfire. So we have a way to support rejecting our null hypothesis that our observed and expected values are the same. It seems like they really are not. And there's statistical support to say that there's a significant difference among them. Lastly, it's nice to graph this data so we can come up with sort of a plot. It helps us visualize these results. And a plot like this is made pretty easily if we simply highlight all of our data here. So we have our independent variable, observed and expected values, and our dependent variable, which is basically counts of them. And we're sort of looking at this independent variable being partitioned into these different factors, which are these different feeding guilds. We can highlight all this data, click on bar plot, insert bar plot, and lo and behold, we get a nice graph. And as before, I would modify the graph a little. Chart titles, I think, aren't very informative. I would take that out. The lines, I don't think are very informative. I would take them out. And then all we really need to do is add a title to the y-axis. And what does the y-axis represent? It represents counts. So we just put counts. And now we have a nice plot. It tells us we have this independent variable of post-fire and pre-fire data, observed and expected categories. And these categories are broken down into birds that tend to feed on seeds, birds that tend to feed on insects, or those that tend to feed on nectar, carrion, or those that are generalists. And we can compare the counts of them between post-fire and pre-fire transect surveys. And so this is a very nice way to take all this data, analyze it, see whether there are some significant differences. We test a hypothesis that there are no significant differences. That hypothesis in this case is rejected. So there are significant differences, and we can graph the data here for visualization. All right, so I hope all of this information has been meaningful, in helping you understand how to analyze data in Microsoft Excel relatively efficiently, so you don't have to do so many calculations by hand. You can do your calculations pretty quickly, and also how to analyze your data using some statistical tests, t-tests, chi-square tests, in order to generate these p-values that allow you to assess your null hypothesis and tell you whether or not you should reject your null hypothesis or accept your null hypothesis, and how we can potentially graph some of this data so that we can present it for the sake of visualization. All right, that's it for this graph, this graph. That's it for this video. Happy stats thinking, folks. I'll see you next time.